This is the third video in cell division. In this video, we will discuss what nondisjunction is and how it can lead to major chromosomal abnormalities which affect the survival of a zygote. Nondisjunction is the failure of a pair of chromosomes to separate during anaphase of cell division, which results in one daughter cell receiving an extra copy of a chromosome and the other daughter cell receiving no copy of that chromosome at all. If this occurs during either meiosis I or meiosis II, it can result in gametes that do not have the correct number of chromosomes to bring to fertilization. In normal meiosis, this diploid cell, with one set of homologous chromosomes, would go through interphase and enter meiosis I, where the chromosomes would replicate in S phase and then line up as homologous chromosomes in metaphase I. This cell would divide, and each cell would then progress into meiosis II, where the sister chromatids would line up during metaphase II, eventually resulting in 4 haploid daughter cells. If nondisjunction occurs during meiosis I, then the homologous chromosomes fail to separate during anaphase I. When these daughter cells of the first round of division go through meiosis II, the resulting gametes will either be missing a chromosome or will have gained an extra chromosome. If nondisjunction occurs in the second round of division, then the homologous chromosomes successfully separate from each other during meiosis I. However, in meiosis II, anaphase II fails to occur properly in one of the 2 daughter cells. This results in one cell that successfully completes meiosis II and forms proper gametes, but the second cell does not, resulting in one of the 4 gametes missing a chromosome and the other having an extra copy. Nondisjunction is problematic because if a gamete does not have the correct number of chromosomes for a haploid cell, then the diploid zygote that forms after fertilization will also have the incorrect number of chromosomes. Using the cells we looked at in the previous slide, a haploid sperm with 1 chromosome should fertilize a haploid egg with 1 chromosome, forming a diploid zygote. Notice that this cell is genetically identical to the one that started the process of meiosis earlier on the previous slides. However, if a sperm cell that did not contain a chromosome fertilized a haploid egg, the resulting zygote could not be diploid. Instead, it contains only 1 copy of the chromosome. When a cell is missing a single chromosome from a homologous pair, it is called a monosomy. Similarly, if the sperm cell that fertilized the haploid egg was carrying an extra chromosome, then the fertilized zygote would have an extra chromosome also. The fertilized zygote contains 3 copies of the homologous chromosome instead of 2. A cell with 3 copies of a chromosome is called a trisomy. Major chromosomal differences involving whole chromosomes or large pieces of chromosomes occur quite commonly, yet are seen infrequently in offspring because they are often lethal. The most familiar chromosomal difference seen in live births is Down syndrome, which is also called trisomy 21 because it is the result of an extra copy of the 21st chromosome. Down syndrome can be diagnosed by looking for the extra chromosome in a karyotype, or a picture of the chromosomes. We can see here chromosome 21 has 3 copies. There are approximately 13 children born with Down syndrome out of every 10,000 live births. The only other 2 autosomal trisomies observed are with chromosomes 18 and 13. However, those infants do not typically survive longer than 1 year. Unlike the autosomes, differences in the number of sex chromosomes, the X and the Y chromosome, is not typically lethal. In fact, individuals who are trisomy for the X chromosome or diploid for the Y chromosome are found among healthy males and females and often do not even know that they have an extra chromosome. An extra Y chromosome often has little to no effect on male development because there are few functional genes that exist on the Y chromosome, other than the gene that stimulates the male development pathway. While the extra X in females does not cause problems because of a process called X inactivation, which we will discuss in Chapter 17. Two other sex chromosomal abnormalities do have phenotypic effects on sexual development. Males with an extra X chromosome have the genotype XXY, called Klinefelter syndrome, have a distinctive group of symptoms seen here. These males do not produce sperm and thus cannot have children. In female, the loss of an X chromosome, also called Turner syndrome, causes the phenotypic changes listed. Because they have ovaries that are small or absent, they do not typically menstruate and also cannot have children.